Let's talk crinoids and their relatives. So, in the beginning, there were not crinoids, there were eocrinoids, dawn crinoids. Because remember, eo means dawn. I think it's Greek or something like that. I don't know. Yeah. And here they are. So, these things have arms. And the arms, we think, have ambulacra on them. Although these are kind of twisty, little helical spiral arms. Yeah. And that's pretty common on some eocrinoids. Eocrinoids is maybe an older term, and it's a term that's applied to kind of a broad range of things. Here's a couple of them from a textbook. These things probably aren't very closely related, because if you look at that one, you'd say, no, that's just like a stylophoran that's lost its stylophore. That's like a full-on crinoid, basically. We're on the way there. And this is like an untwisted version of a helicoplacoid, but with arms? Yeah. So hard to know exactly where to put these things, and it may be that we need some earlier fossils of some of the forms that lead up to these in order to figure out how these go together. Because it looks like there were probably really small genetic tweaks that could happen in an echinoderm genome that did not impact the survival of the organism, but made a real difference in the shape. Because they're all making plates. The plates are all coming from the middle layer of their body, but you stretch the plates in different ways, and all of a sudden you got a very different looking creature. These are hard to reconcile. But if you look at them, they've kind of got a cup up here, and they've kind of got this sort of hollow trunk. And this was sometimes, we think, rooted to the ground. Sort of the way a Helicoplacus or Helicocystis or an Edrioasteroid was rooted to the ground. So you can think of it as like a stretched out Edrioasteroid that has now got, instead of just ambulacra running on the top, it's usually got 10 little arms that are going to be sticking up. It's going to lift its little ambulacra up off of its cup. And yeah, maybe that was easier to do genetically than it seems, because it seems like a big deal sitting here right now. But yeah, we don't know how the early echinoderm genetics really worked. So here's another thing that is put in with the eocrinoids. I would tell you it's really kind of different from the eocrinoids, but it's classified with them for now, and we have some really good fossils of them. We also have fossils of some Gaugia as well. Those are pretty common out in Utah. And these are from Morocco, and my one is not colored like this, although they may have added some of that coloring. I'm not sure. To make it extra beautiful for sale. Or maybe that's just how it comes out of the ground. Hard for me to tell. But yeah, Ascocystides, and here's an Ascocystides. This is like an external mold of an Ascocystides. Here's one where you can actually see all the plates, or maybe this is just where they push clay into the mold so that you can read out what the plates looked like. Again, there was a body on the outside of this, at least some kind of skin, and that is gone, 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 as are the internals. So we think the cup is for digestion and excretion. And so this one is a lot closer to being a blastozoan, like one of these sort of cup and stalk animals, like a modern crinoid. But it's not classified there at the moment. It's another thing in need of revision. So there you go. Okay, next step up, and this one's annoying because it's really hard to define precisely. Rhombiferans have lots of little, they're not really arms, they're more like pinnules, which is like a little version of an arm, but it doesn't have an ambulacral groove of its own running up there. It's more like it's an armored tube foot, is how I would think of it. Which would be like you would get on brittle stars. They have all their tube feet are armored like that. So here's some dead ones laying on the ground. They've been slaughtered, or maybe they just live on the ground. This one's dead for sure. It's just like the skeleton. And here you can see the plates on these things quite often, I think, resemble the Union Jack, the British flag with the red, white, and blue, as they call it, or blue, white, and red. I don't know. Maybe that's the French. But in that sort of X and cross pattern. Yeah. And so it's just printed on the plates. And so presumably some of that transferred to the outside, and you wouldn't be able to see that if you could see one that's alive. Now, this thing that's underneath it, again, it's not a stylophore, but It's curling with it. It's probably got muscles inside of it. It may be descended from a stylophore, or it may be serving the same sort of function. It may not be like it came from a stylophore. It may have come from a common ancestor with all the things that have stylophores on them. Yeah, echinoderms. Yeah. Wow, what a mess. Yeah, but they're really cool looking. Look at that. And these used to be just all over the place. If you go up to Oklahoma in the Ordovician there, you'll find lots and lots of these little plates, just all around, little Union Jack hexagon plates. All right. Next group of annoying creatures are the Diploporita. They're named because they have two openings in there, and so the idea is that this allows extra respiration. Maybe one of them is probably doubling as the mouth, but you can also, when you're taking in seawater or whatever, that you can use the oxygen from that theoretically. And here's our two pores that are over here. But this is probably a group that is paraphyletic. That's what the latest studies are showing. And these are studies that are based on grouping things by shape, by how they look. So, but it looks as though this idea of having two separate big openings, like pores in the top, may have been invented several times by several different groups. So here we have like the earliest crinoid, and so its mouth is in the center, and this extra opening is over here, which kind of looks like an anus to me. Yeah. And here we have a diplopore, one of these diploporites, and mouth in the center, little anal pyramid down there. And even their arms are very similarly structured. So these would have arms coming off. They're broken off of these specimens. But you can see they would have carried on right here and here and here and here and here. So these ambulacra would have connected out to arms. The arms are a continuation of the ambulacra, so the mouth parts are right up here. The ambulacra are the yellow bits that are coming down. And they both have little covers over the top that can open and shut. And in the earliest crinoids, it looks a lot like the structure you'd see in diploporites, with one big plate that's got a little notch out of it on the bottom. And you can find these at several of the places that we will go to. Crinoid arms are not cylindrical. They would look similar to the crinoid stem plates, but they would be sort of U-shaped profile. Yeah, like that. So yeah. Diploporites. We've got a couple. We've got, it's not quite this one, but it's a really close relative of it. And you can look at it, and it's amazing and horrendous and wonderful all at the same time. Yeah. So enjoy. Yeah. Okay. So rhombiferans and diploporites are both pretty close relatives of paracrinoids. And paracrinoids are this group that are definitely not crinoids, but they're doing a lot of crinoid-like things. So they're blastozoans. So they've got this cup, and all their organs are in the cup, put all their eggs in that one basket. But they do have a stalk. The stalk is not a stylophore, although in some cases we know it does have muscles and it can move, like under control of the brain. Brain's usually like a ring shape that's found, and it kind of goes around the mouth. That's pretty common in these lower life forms, as we can call them, being humans and being the ones running the class. So yeah, too bad, lower life form. Yeah, take that. Here you can see the mouth parts, and it's not at all symmetrical, but it kind of gives the impression of fivefold symmetry while being completely asymmetric. But yeah, you got some sort of developmental gene patterning that's going on here between these different groups. And these plates in the center are presumably covering the mouth. So these darkened ones in here are the mouth covers. And the plates that are running up here are probably going to be ambulacra covers that can open and shut, as with the mouth plates. So it's nice. They can shut their mouths by shutting a little plate over their mouth, because their mouth is just a hole, like our mouth is a hole. But we have something that we can shut over our mouth hole as well. So good for us. Ours doesn't have bone in it. Theirs did. Slightly different, but not that different. Yeah. All right. Here are some paracrinoids. Paracrinoids. Oh, annoying. Look at this. This is a mouth. These are two ambulacra. This is a mouth right there. This one comes from Oklahoma. And we have several of this, some that I found. Actually, all that I found. And look, it's got 3 ambulacral grooves. Here we've got what looks like 5. Here we've got another one that's got 2, basically, going either side of the mouth. And there are also some that would have what looks more like 4 coming out, which I think is what we got here. We got it in splitting here. 1, 2, 3, 4. So 2 or 3 or 4 or 5 ambulacra all feeding into the mouth. It's like, how many conveyor belts do you need feeding your mouth? You know, whatever is optimal for the environment where you are, or whatever is good enough for the environment where you are, because good enough will get you by, and evolution is a lot more based on what's good enough than on what's perfect, I would say. Now, all the paracrinoids that we call paracrinoids do seem to be more closely related to each other than to other things. And look at this. This is the ambulacra. So we're coloring the branches. Every purple branch has 2 ambulacra. And yellow is going to be 4, pink is going to be 3, and orange is going to be 5. And so you can see that the number of ambulacra is not very well conserved. So you got this group. It's like purple, purple, purple. So you'd say, oh, this is like a 2 group. And then right in the midst of that, coming off of here, is a 5-sided ambulacra group. Oh, okay. This little lineage starts with a 2 and then has some 3 over here and has a little 4 going down here. Doesn't ever quite get to 5. Can't count that high, I guess. But what you're possibly looking at is a pattern of development where the early versions start with 2 ambulacra and then they figure out how to bifurcate those 2 or grow another one in the middle of those 2, which is like bifurcating at a different point. You're just bifurcating at the point where you have 2 and you're just bifurcating one of them. Because, echinoderms do not follow your petty rules of symmetry. Yeah, as if. Scientists have invented this thing called Loven's law, and it's the rule of 2-1-2 development. So in echinoderms, what you'll see is that it's not 5-fold symmetry. And if you look at the plates and the order in which they develop in the developing organism, it's going to go 2-1-2 at the early stage, rather the darker green, then 2-1-2. And so with 2-1-2, if you just don't have an ambulacra on top of this, you've got either 2-fold or 4-fold symmetry. If you have this one and you don't have a separate, if you just sort of close this off and close this off, then you'll have 3 ambulacral symmetry. If you don't express this one but do express both of these and bifurcate them like that, then you'll have 4 ambulacral symmetry. And if you run ambulacra off of all of them, you'll have 5. And is there any reason why you can't bifurcate these even more? Not really, but I don't think they did. So, yeah. Although there are some starfish now that have many more arms, and there's ambulacra down all those arms, so maybe starfish are the great experimenters. Yeah. Okay, next in blastozoa. These are blastoids. Blastoids look just like this every single time. They have a stalk. They may or may not have a holdfast in life. That is debatable. A lot of their relatives do, but some of their relatives don't. The little sort of side branches that come off the stalk are called cirri, but it's like a little stalk off your stalk. Yeah, adorable. And these are all motile. They have muscles and nerves and stuff running down these columns, so they can potentially move them, although it may not be a particularly important part of what some species did. But like, it's baked into the design that this is like the old stylophore, which was for flipping side to side when you need to get around. And it's just a question of how much of that has been lost in some of these different groups or retained. So these are going to have a mouth, a separate anus, and quite often they will also have a gonopore on the top. And the gonopore is going to be where all the little eggs and sperm are going to come out. Yeah. So we think these things in life had 2 little rows of essentially armored tube feet. These are these brachioles. It's like a covered, I believe, segmented tube foot. And so these would have been involved in capturing food and feeding it up the ambulacral groove in toward the mouth. Blastoids tended to be found, they're super delicate, we think, in life, and they tended to be in really, really sheltered lagoons where you'd find a lot of other really delicate things. So if you're thinking big, robust clams that get, pounded by waves and do not care, those are not found in the same places as blastoids. Because if there were blastoids there, they would get blasted, essentially. Yeah, not as tough as they look. Yeah. Okay, and we made it to crinoids. Congratulations. Crinoids, these are our little sea lilies like that. And here's a beautiful picture of several of these that are kind of attached themselves to a piece of fossil wood down here. It looks like a carving in the rock, but it's actually a carving around all the fossil elements. It's very nicely done, but it's done in a way that almost makes it look fake. Yeah, but they're real. They're the real deal. Get used to crinoids, because if you go fossil hunting in the Paleozoic, you will find crinoids. And you won't find all of the crinoid, because, look how much stem they have. And then this little thing right there, that's the cup. The cup tends to be very difficult to find, and when you do find it, it'll often just be one or two plates from a cup that have broken apart and fallen all over the place. But there's lots and lots of these little cylindrical plates that would have made up the columnar elements is what they're called. And those are super common. There are large places where all the limestone is just pressed together crinoid columnars. They're marvelous, and we call that crinoidal limestone. This is what a cup looks like if you ever find one. This is a cup that has some of the arms still on it, but I wanted to give you a picture of the whole thing, because when you're looking at the fossils, you're going to be looking at this thing and thinking, how was that alive? What did it do? It's just a little segment of the stem of this big long thing. And so the flesh would have been running down the center, presumably maybe some little water vascular system, definitely some muscles, so it could move around these little parts. And it would have been covered over in a bit of a skin. Yeah. All right. And if you were to break a crinoid apart and dissect its little innards out, which is a mean thing to do, shame on you, then you would find something like this. So there's a mouth that's going to come around, and then boop, there's the little anus. So food goes in, food goes out, kind of in the same spot. The problem for this is that snails find crinoid poop just irresistible, certain kinds of snails. And so you will often find gastropods that are clamped over the snail's anus. And the problem is that the anus is right next to the mouth. And so if you're clamped over the anus, waiting for the good stuff to come out, you can actually prevent stuff going into the mouth, and that can actually just kill the crinoid. And the gastropod doesn't appear to care. It just moves on to somebody else and, murders another one. But it's, pretty bad. And so in later crinoids, you will see they develop some, sometimes they'll put the anus up on this giant, long sort of projection, up in the sky. They call it an anal chimney. And that's just to get it away from the mouth. So if something bad happens to the anus, then there's no chance that the mouth is also going to be blocked. And you'll also see a lot of crinoids that'll develop big, long spines that they can control, so they can wave them around like a little, get back sort of stick. Hey, stay away from me. Both around the mouth and around the anus. And so we'll get to those later. Yeah. But if you're looking at the arms of the crinoid, the yellow bits are the parts that are actually stereoms. You have one big sort of curving, kind of bean-shaped plate on the bottom, two little plates on the top, and there's your curving sort of semicircular plate on the bottom, and your two little plates that cover. And then all the way on the top, you'll have the tube feet. And in the early ones, those tended to be covered by their own little stereom plates that were probably closable and openable. And in the more modern ones, they tend to just be covered in basically epithelium. But sometimes they can close all their arms together, like in that fossil. And when they're doing that, they've essentially armored up around the outside, because the bottoms of the arms are pretty thick like that. So that's crinoids. That's how they work. Okay, we'll come back in the next and I believe final section and cover the modern ones, and then a little bit more of kind of how they work, what they do, so you understand them. Yeah. Because understanding is the first step in love, isn't it? Sure. Yeah. Echinoderms are pretty cool.